Laboratory diagnosis of malaria. Before we will move to the laboratory diagnosis, we go through the life cycle and some other information. When we consider the malaria infection, it is caused by the Plasmodium species. When we consider Sri Lanka, Plasmodium vivax is the most common parasite cause malaria. The second one, Plasmodium falciparum. Other two, Plasmodium ovale and Plasmodium malariae. These two are not present in Sri Lanka. If we detected these two species, we have to get from the travelers. That means the travelers they goes to the prevalence or endemic area, and if they come back to the Sri Lanka, we can identify. But most common one, Plasmodium vivax. When we consider the life cycle, there are two hosts they need. One is the mosquito, other one is the human. In here, the definitive host is the mosquito, Anopheles cutis species. Second intermediate host is the human. When we consider first the mode of transmission, they are transmitted to the human by the biting of infected mosquito, female mosquito. When they biting, it is the sporozoid enter into the human body and it is lodged into the liver cells and it will multiply. Here we call sky zone, and inside the sky zone we can see large number of merozoites, that means more than 10,000. When the liver cell or hepatic cell is fully filled, it will rupture. Then these merozoids release into the bloodstream and they infect the red cell. So from entering the sporozoid to the rupture of the merozoid sky zone in the liver cell, up to this we call extra erythrocytic cycle or liver cycle. After rupturing, the merozoid are released and the merozoid enter into the RBC. So this is cycle we call the erythrocytic cycle. Here we can see there are some morphological changes. The merozoid enter into the red cell and it become a ring shape. So we call ring form. Then this ring form converted into the tropozoid form. In here we can see the malarial pigment inside the cytoplasm of the parasite. Because they are start to eating or take the hemoglobin from the red cell. Then this tropozoid converted into sky zone, and here the nuclear division is occur, that means increase the number of nucleus. So we can see a species to species number of nucleus. Here we call sky zone, and inside the sky zone we can see the merozoid. So in the liver cycle also we already discussed merozoid. Here also we discussing merozoid, but in the case of liver cycle we can see more than 10,000. If we want to see this, we have to take the liver biopsy and we have to go for the HME staining. But here by using Romanesque staining like a Leishman stain or Ginsai stain, we can detect. And then the sky zone ruptured, and if it is fully matured it will ruptured, and this merozoid enter into the red cell and it become gametocyte, male and female gametocyte. Then this male and female gametocyte, where it is present in our bloodstream, then again the mosquito they take the blood meal. These gametocytes enter into the mosquito and it become a female gametocyte converted to female gametes, male gametocyte converted into male gametes. Inside the mosquito there are some morphological changes, but In the case of female gametocyte, it converted to female gametes, that means it is matured. But in the case of male gametocytes, it's converted into male gametes, that means normally we all know the shape of the gametocyte, round shape, but in the male it converted into again a spiral, the spheroid shape, right, the elongated shape, and it the fuse with the female gamete is its form zygote, and it's continue the life cycle, and it's produce the spheroid. Right, this is the life cycle. When we consider 4 species of Plasmodium species, in the erythrocytic cycle or RBC cycle, we can see the different morphology. When we consider the Plasmodium falciparum, first we will discuss that one. Normally in our peripheral blood we can see the ring stage and the gametocyte. The other 2 stages, that means tropocyte and schizone, normally we cannot see, but if it is severe infection only we can see the tropocyte and the schizone. Another thing, we can easily identify the falciparum in blood smear, because in the case of ring stage, or we can call early tropocyte stage, in here in single red cell we can see the single ring stage, or sometimes we can see the multiple rings, that means more than 2 we can see, or sometimes we can see the coliform. Coliform mean edge of the red cell we can see, or sometimes we can see double dot ring form, that means in this stage we can see in the 2 nucleus. Then late tropocyte or normal tropocyte we cannot see normally. If it is severe cases only we can see. Then next step is the schizone. This also normally we cannot see in falciparum species. In severe cases only we can see in these stages. Then gametocyte, male and female gametocyte. How we can differentiate these gametocyte from other 3 species? This is significantly morphological differences we can see. That means the crescent shape or banana shape. In the case of female, we can see crescent shape, that means pointed the cytoplasm. In the case of male, rounded cytoplasm. When we consider the color of the cytoplasm of the parasite, in the case of female, that is blue color, and the nucleus very dark pink or red color. But in the case of male gametocyte, the cytoplasm is the bluish pink color, nucleus pinkish blue color, and diffuse. This is diffuse. But in the case of female, this is compact. That's why the color is dark in the case of nucleus. So we all know the features of the falciparum, Plasmodium falciparum, erythrocytic stages. Next we will move into the Plasmodium vivax stages. This is Plasmodium vivax is the most common one present in Sri Lanka. So we should know how to identify the Plasmodium vivax. Here when we consider the Plasmodium vivax early tropocyte or ring stages, here we can see single ring inside the RBC, and it converted into tropocyte or late tropocyte. The cytoplasm of the parasite, the shape is not clear, but in the case of early ring stage we can see very nice ring like cytoplasm. But the late tropocyte or tropocyte stages, the cytoplasm of the parasite, we cannot see the exact shape. Then these tropocyte stages converted into the schizone. Here in the case of schizone, nuclear division only happened. So this schizone ruptured and released the merozoite. This merozoite entered into the new RBC. Its form gametocyte, male and female gametocyte. Here the gametocyte, when we consider Plasmodium vivax gametocyte female, this is round shape, and when we consider the nucleus of the parasite, this is dark red color and compact eccentric. That means eccentric means one side we can see seen, and again the color of the nucleus is very dark color, compact. When we consider the cytoplasm of the parasite, blue color. Inside the cytoplasm we can see brownish color, yellowish brown color, malarial pigment. The malarial pigments are present inside the cytoplasm of the parasite. When we consider the red cell, inside the red cell cytoplasm we can see the Suffner dots. And when we consider the morphology of the RBC, in the case of Plasmodium vivax, morphology is larger and enlarged RBC compared with normal RBC, and the color is pale color compared with normal one. When we consider the male gametocyte, it's also the red cell morphology. When we consider the red cell morphology, enlarged RBC, pale color. When we look at the morphology of the gametocytes, it's also round shape. The cytoplasm color is bluish pink color. The nuclear color is pinkish blue. So this is diffused because the chromatins are diffused. So that's why it's get light color compared with the female one. That's all about these two. When we consider other two, Malaria and Ovale, now we will discuss the Ovale. The Ovale, when we consider the red cell morphology, the red cell, infected red cell is oval shape. In the case of early trophozoite stage, when we consider the parasite, the cytoplasm of the parasite very thicker. This is the significant characteristic of the Ovale. Then when we consider the late trophozoite stages, again cytoplasm of the parasite is thick compared with other three species, and the red cell fabricated, that means broken RBC. Again when the late trophozoite converted into sky zone, here also red cell, the oval shape, and the fabricated, that means broken RBC. And when we consider the sky zone, there is a shape, but in the case of vivax we cannot see the actually shape of the sky zone, amoeboid shape. But here in the case of Ovale sky zone, there is a shape. When we consider the female and male gametocyte, the parasitical morphology is similar to the Plasmodium vivax gametocyte, male and female, but red cell morphology somewhat differ from the Plasmodium vivax and the Plasmodium ovale. That means in both cases red cell enlarged, that means enlarged, but in the case of ovale, the red cell is, infected red cell is oval shape or fabricated or broken. That is the significant characteristic or significant differences. When we consider the Malaria, the early trophozoite is very smaller, but the cytoplasm thicker compared with the vivax. And the late trophozoite form we can very clearly differentiate other three species. This is band shape. And the sky zone stages also there is a shape, rosette shape, and the case of female and male similar to Plasmodium vivax. Now you all know how to differentiate the Plasmodium vivax, Plasmodium falciparum, Plasmodium malaria, Plasmodium ovale. Now why we want to differentiate these four species? Because in the case of Plasmodium falciparum, they produce, that means compared with other three, they are the very dangerous ones. Sometimes it will lead to death also, because they are infect the brain. It's called cerebral malaria. So we have to identify species wise and confirm, and we have to give the treatment. Now we will move into the laboratory diagnosis. First we have to do the microscopic test. Here we have to take the peripheral blood smear. This is the gold standard method. Normally we have to prepare thin and thick film. We can prepare thin and thick film separated slide, or sometimes we can prepare it in single. By using single slide we can prepare thick and thin blood film. In the thick film, when we consider thick film, more sensitive, because we are adding more than 1 drop, nearly 3 drop of blood, and we are preparing thick film. So here we concentrate the parasitical stages, so it is more sensitive. Thin blood film we can identify, mainly we are using for the species identification. Next one, fluorescent microscopy we can use. Other one, quantitative Buffy code examination. Non-microscopic tests are antigen detection by using ICT. Other example, the antibody detection by using ELISA. We can culture it, culture technique, then molecular diagnosis. Then when we look at the peripheral blood smear, this is the gold standard method. Normally we have to take the peripheral blood, we have to collect, and we have to prepare the smear, the thick and thin blood smear. Type of peripheral blood smear. Normally if we want to identify the malaria parasite and species, we have to prepare thick and thin smear. Sometimes we can prepare thick and thin smear separated slide, or in same slide. So if we prepare thick film, before go to the staining step, we have to dehemoglobinize, because our whole blood, when we consider our whole blood, it's contain high concentration of RBC, red cells. So when you are staining without removing the RBC, the RBC get the stain, so we cannot detect the species or parasite. So first we have to do for the dehemoglobinization. So if you prepare in thin and thick film on the same slides, first you have to do the thick and the dehemoglobinization carefully, without disturbing or without mixing of the distilled water in thin film, because if any water drop come to the thin film, the red cell morphology will damage. So if we want to identify the species, we need the red cell morphology. So carefully we have to dehemoglobinize the thick smear. Then we have to go for the staining step. So you all know how to prepare the thick and thin blood film. So by using thick blood film, we can increase the sensitivity, so we can identify, or we can tell the patient is positive for the malaria. But if you want to identify the species, you have to examine the thin blood film. Then we will move into the staining step. We can use Romanesque stain, such as Leachman stain or Giemsa stain or field stain. So when we consider the Leachman stain, we can directly add the Leachman stain particular amount, for example 8 drops. Then we have to keep 1 or 2 minutes. Then we have to add double amount of distilled water, and we have to keep 5 to 10 minutes. So here this is the step we have to the staining. But in the case of Giemsa stain, before we adding the Giemsa staining, we have to do the methanol fixation, because In the case of Leishman stain, we are preparing Leishman stain in the methanol. That's mean we adding the Leishman powder into the methanol, concentrated methanol. So it's contain methanol. Normally methanol act as a fixative, but in the case of Giemsa, when we preparing the stock solution, we adding small amount when we preparing the Giemsa stain, the Giemsa powder we are adding to the methanol. Then when we in the stock solution, when we are preparing the working solution, we are diluted in 10 times for the working solution. So the concentration of the methanol is very low, so the water content is high. Sometimes if you are without fixation, the red cell morphology may damage. So before the Giemsa staining adding, so we have to do the methanol fixation 1 minute, then only you can go for the staining step. This is the difference between the Leishman stain and the Giemsa stain. So the advantages of the Leishman stain, directly we can add the stain and we can go for the staining step, but in the Giemsa stain there are extra steps that we have to fix with the methanol. Then advantages of the blood film. So it is very its preparation is simple, rapid and cheaper. Then the uses of the thick blood film, we can detect the parasite. Actually 40 times more sensitive than the thin blood smear, and we can quantify the parasitemia. That means we can count the number of parasites by using that, whether the patient is very severe or less severe. And here we can demonstrate the malarial pigment by each and every species color. Malarial pigment color may be changed. So the thin blood smear, normally we are using to differentiate or identify the species by using the morphology of the RBC. We already discussed the morphology of the RBC and the species earlier. Disadvantages here, the thing, actually blood smear, it is labor intensive. That means it's take more time. Other thing, if you want to identify accurately, the technician should be, the microscopist should be very experienced one. Other thing, if the smear is very lower sensitivity, so we have to detect. The other thing, in the case of thin film, if it is low sensitivity, low concentration, difficult to detect. Right, species identification we already discussed. Right here you can see the actual picture of the parasite. Here the falciparum species, different the stages you can see. The first picture you can see the multiple ring stage and a coliform. The second one you can see the double chromatin stages. Then third one you can see the gematocyte. I think this is the female gematocyte. The pointed end under the nucleus, very compact. Right, next one, this is Plasmodium vivax. First one ring stage you can see, second one gematocyte, third one schizone. Here the Malaria, the band form, that means late tropozoite stage. The second picture explaining the Plasmodium ovale, the ring form. Then quantification of the parasite. Normally why we need to quantify, especially in the case of falciparum, we have to quantify. We already discussed it causes the cerebral malaria, so we have to quantify. Then the fluorescent microscopy, here we have to use the fluorescence stain, and we have to identify. Here we are using acridine orange, the fluorescent dye. Right here you can see the actual picture of the parasite. Here the falciparum species, different the stages. Right, next one, this is Plasmodium vivax. First one ring stage, you can see. Second one gametocyte. Third one schizome. Here you can see the qualitative Buffy-Coat method. I don't need the procedure. Thank you.